Hello all, we'll discuss about RNA editing now. RNA editing is a process in which the sequence in RNA is changed after the transcription takes place. This RNA editing is actually an altering process so that we will be getting a protein that is entirely different from the gene sequence that we have predicted. This RNA editing can be either insertion, deletion or changing of the nucleotides which composes this RNA. And there are two major mechanisms with which this RNA editing is taking place. And the first one is deamination, second one is guide RNA directed RNA editing. First, we'll start with the deamination. Deamination is applicable to two bases, cytosine and adenine. This cytosine is deaminated to form uracil that gives a different base, and this causes an alteration in the RNA sequence. And this is also applicable for adenosine. Adenosine is also deaminated to form a inosine base that can base pair with cytosine. And here, cytosine deamination is shown. In this cytosine deamination, we take an example, that is mammalian apolipoprotein B. This mammalian apolipoprotein is actually responsible for the transportation of lipids, but this RNA, it gets altered when it goes to different tissues. So you can see the pre-mRNA with which both exons and introns are present, and you will get the mature mRNA in which only the exons are present. And there is a codon shown CAA which codes for glutamine. And this mature mRNA, when it reaches the liver tissue, there is no change or alteration taking place, and it codes for glutamine and gives a protein of length 4563 amino acids. And it is responsible for the transportation of cholesterol and triglycerides. And when the same protein, that is the same protein, it gets altered when it reaches the intestine, the alteration is deamination or site-specific deamination of cytosine. And the cytosine is deaminated to form uracil or uridine. This uridine residue, when it forms, it changes the codon into UAA. Instead of CAA, you have UAA in the position, and this UAA is actually stop codon. So whenever the ribosome reaches this UAA, recognizes it as a termination codon, and the process of translation is stopped there. It will be getting a protein of length 2153 amino acids. Okay, so the same process, that is, you are using the same mRNA transcript, but when it reaches the two different sites, the alteration is taking place. In liver, there is no alteration. You'll get a protein of 4563 amino acids long, and the same protein or same RNA, when it reaches the intestine, due to the deamination of cytosine, you'll get the UAA or stop codon. You'll be getting the protein of 2153 amino acids length. And this shorter protein is also responsible for transportation of dietary lipids, but it is actually taking place in the intestine, different from that of the liver. So this is an example for RNA editing. Adenosine deamination. Here the adenine nucleotide is getting deaminated. You will be getting a nucleotide called as inosine, and this inosine is different from adenine because it can base pair with cytosine. Usually adenine base pairs with thymine, but inosine base pairs with cytosine, which entirely alters the sequence of this RNA. And this also causes certain changes, and an example for this is the protein that is getting altered in mammalian brain. It causes the alteration in the permeability of calcium channels. So normally you will be getting a protein which is present in as adenine, but when it changes into inosine, it base pairs with cytosine, which is an unusual base pairing, and causes the sequence alteration in the RNA. Again, you are getting a different protein that is different from that of the gene that you are predicting. So these are the two deamination process. First one, cytosine is deaminated by the enzyme cytidine deaminase, which gives uracil. When it becomes uracil, it becomes a stop codon, and the translation is stopped there. A shorter protein is obtained. And in second case, you can see there is a adenosine which is getting deaminated by the enzyme ADAR enzyme. This enzyme is responsible for converting or deaminating adenine into inosine, which base pairs with cytosine. Second mechanism is the RNA editing by guide RNA. Here you are using a different RNA which is called as guide RNA, that is being entirely transcribed or differently transcribed from a different gene which is having no relationship with the mRNA that we are using. Here the guide RNA is synthesized, or which is being transcribed, having 40 to 80 nucleotides long, and this particular guide RNA is used for the editing of the RNA that you are going to be getting altered. And this happens in the mitochondria of certain trypanosomes, especially in the COX-2 gene of trypanosomes. Here, the RNA editing is taking place by guide RNA. Guide RNA is actually used for the insertion of certain residues, especially uridine residues in the unedited RNA, which causes the alteration in the gene sequence as well as in the shifting of the frame. So whenever the ribosome is reading during the translation, it counts the codon in numbers of 3, but whenever a single or 2 or more uridine residues are introduced into this particular codon, that causes the codon to get changed, as well as the frame is also getting shifted. So the entire sequence or the entire information is being changed due to the addition of this uridine residues. And this guide RNA, which is 40 to 80 nucleotides long, is consisting of 3 regions divided as anchor region, editing region, and polyuridine rich region. This anchor region is actually somewhat near to the 5' end and consisting of certain sequences that helps to direct this guide RNA to the unedited RNA. The second region consisting of the editing region. Editing region is mostly composed of adenine residues. In opposite to this adenine residues, the uridine residues is being added to the targeted RNA, and the 3' end, towards the 3' end, there is polyuridine rich sequences, polyuridine rich sequences that are important or that are helping in base pairing or interacting with the purine rich sequences in the unedited RNA. So these are the 3 different regions in guide RNA: anchor portion, then editing region, and the poly-U rich region. Here, this guide RNA, the anchor region directs the guide RNA to the unedited RNA. They undergoes base pairing, and the editing region, which contains adenine residues. This adenine residues is actually does not having any complementary bases in the unedited RNA, so they undergoes looping out. Okay, so since they do not have any complementary bases, they always undergo a looping out process, and you can see that they are as looped out. Normally, not like a linear sequence, but loop out can be seen in this guide RNA, and they form a RNA duplex in which there is a targeted RNA and guide RNA is present, and in this guide RNA, wherever there is no complementary bases, like there is no bases opposite to the adenine residues, those adenine residues will loop out. And what happens is that recognizing this looped out position, certain endonucleases will come, and this endonucleases will cut at the targeted RNA at positions where this looping out is taking place. So you have different looping out positions wherever these adenine residues are present, and wherever there is no complementary basis to these adenine residues are present, this endonuclease is cut at targeted RNA opposite to these adenine residues. And after that, uridine residues are added to this gap positions. Here you can see that there is the guide RNA and the targeted RNA, and the site of U insertion is also shown. Here, whenever there is a looping out position, at this loop out, endonuclease will cut at this mismatch, and to this position, uridine residues are being added. This is actually performed by an enzyme TUTase, and uridine residues are added at different positions. And after the addition of these uridines, this ligase joins the Nick, which is formed in between these nucleotide bases, so that the RNA, that is a targeted RNA, the structure will be complete. But the thing is that it entirely alters the codons of this RNA and also causes the frame shift. So you are getting an information in the RNA which is totally different from that of the gene sequence. So this is how guide RNA directed RNA editing takes place. So this is another picture which is showing the same, and you can see that there is a change undergoing or in the amino acid. The codon changes, the amino acid also changes. That causes a different composition in the protein. And that's all about the RNA editing. So in RNA editing, what we said is that here it is an altering of the RNA sequence after the transcription, but this is different from that of which is taking place in the gene sequence. So you are getting a protein that is different from that of the gene that you are predicting, and it having 2 different processes. First one is deamination, and the second one is by guide RNA directed RNA editing. Deamination is applicable to site-specific cytidine and adenosine residues, and the guide RNA directed RNA editing is actually performed using a guide RNA by the insertion of uridine residues. Thank you.